Okay, maybe we can go ahead and get started. I'll just ask everyone to mute themselves during the presentation so we don't have any like rogue sound. Okay, well, I will introduce Dr. Perez-Jaramillo. Welcome to this week's Plant and Soil Science Raymond Seminar. My name is Bailey Kretzler. I'm a first-year PhD student in the Crop Genetic Heritage Lab, and it is with much delight that I welcome our guest speaker, Dr. Juan E. Perez-Jaramillo. Dr. Perez-Jaramillo was born in the municipality of Ereco, province of Antioquia in Colombia, and he completed his undergraduate studies in biology at the University of Antioquia. He was later awarded an International Talent Fellowship from the Nevada's government in Spain to complete his master's in environmental agrobiology at the Public University of Nevada. Under Dr. Pedro Aparicio Tejo, he studied the effect of organic matter amendments and more specifically treated sewage sludges on the enzymatic activity of soil and N2O emissions. He was then awarded a PhD fellowship from the Colombian Department of Science, Technology, and Innovations, allowing him to conduct his doctoral research under the supervision of Dr. Jos Rijmakers at the Netherlands Institute of Ecology. In March of 2019, he received his PhD and almost simultaneously started a position as assistant professor of biotechnology at the University of Antioquia, where he now teaches and does his research, coming back full circle on his academic journey. Today, he will speak to us about the microbiome of wild plants, a topic which he has worked closely on in recent years. And without further ado, I will hand things over to Dr. Perez-Jaramillo and let him share his screen.
Well, thank you very much, Bailey, for the introduction. First of all, I would like to thank Bailey for this invitation. I am very excited to see this huge amount of people with not Spanish names, because when I give my lessons, all the names are from Colombia, obviously. I am very happy to be here, and I will share with you mainly my PhD research, and I will talk a bit also about what I'm doing right now. Okay, so I will share my screen. Let me just give me one moment, please. Okay, are you observing my? Okay.
Yes.
So I will present now. So I gave this talk. The name, the rhizosphere microbiome of wild plants. I have to be clear that I will not talk about wild plants in general, but regarding wild plants that they have also a domesticated counterpart, so commercial plants or at least edible plants. Okay, for wild plants there is a lot of information also from the group of Noah Feeder, but I didn't work with that kind of plants. So I would like to tell you where I am. So this is Colombia. This is we are located in the corner of South America, in the north corner, and in red you can see my province. My province, the name of my province is Antioquia, and Antioquia is divided in several sub-regions. And right now I'm working for a university that has a very nice philosophy. So if the students cannot go to the university, I will show you where Medellin is. Medellin is here. So the biggest campus of the university is in Medellin. But the university said many of the students cannot come to Medellin to live and to study in Medellin. So the university went to the regions. All these color parts of the map are the different regions of the province, and make universities in there. So I work for the name of the dependencies, Dirección de Regionalización. So like the regions professors. I am one of those professors that goes to all these regions in Antioquia, And this is my main university is in one of the mountains here in a small town, and facilitating that the students can go there and doesn't need to go to the main city. So that's my university, University of Antioquia. I work as an assistant professor since 2019 in the Institute of Biology, and I will go directly to the point now. So I heard from Bailey that there is the audience is quite broad, quite multidisciplinary. So I will introduce concepts new for some of you, but for others are quite known. So I apologize to those that this is a bit repeated. So when I started my project, I remember that the professor Rijmakers told me, what do you want to do for your PhD? Just like that. And he gave me a few options, and he told me there is a project with common bean. Common bean is one of the most important crops in my region. So immediately I said, oh, common bean, of course. And the work, or the project at the moment was based on Vavilov's centers of origin. Vavilov was a very important scientist from Russia, no, between the two world wars, and he was a very good scientist who has at the very beginning he was very prestigious in Russia, but then he started a fight with Lysenko, and Lysenko was from the side of Stalin, and then he suffered a lot after the Second World War. But he established what we call the theory of the centers of origin. He collected a lot of seeds all around the world, and he started to say that most plants were domesticated, were origin in some specific parts of the world, and then they were domesticated in where they were origin. No, so following these Babylon's centers of origin, it is known now that most of our edible plants have an origin. I have to be very careful here because there are a lot of biogeographists that are not agree with this type of classification, but for the purpose of this talk, we will follow this classification of Vavilov's. So we are located, we are going to be talking a lot about South America and Central America, because one of them, for us, at least for us, one of the most important crops, which is beans, but also in the Andean region, we have potatoes, we have quinoa, we have tomato, and Mexico for sure is the origin center of many species in one valley, which is Oaxaca Valley. And we know now that there are hotspots of domesticated plants and wild plants that were domesticated. And I like to see, I like to make this comparison. So we have here in orange, we have the centers of origin of most of the products that we consume today. So we have in Oaxaca, in the Oaxaca Valley, we have Andes and Amazonia, we have several regions of Africa. Anyway, but we have also some yellowish areas, which the name is the most productive agricultural areas of the modern world. So apparently there is not a match between those two areas. No, the centers of origin are not precisely those areas where the products are produced. No, but in 2004, I think the answer is very easy, and it is because those areas match with the nitrogen deposition. No, so where we put more nitrogen, those areas are more productive. So it is normal that China, India, Europe, or a huge part of North America are the most productive because more nitrogen is applied in there, and doesn't have anything to be, is not related with the center of origin. Yeah. So I will talk a bit about plant domestication. just a bit. I am not an expert on this. Maybe Eric can help me a bit if I am saying something wrong, but plant domestication started with gramíneas, no, with this. How do you call it? I remember the name in English, but in Spanish we call gramas. Yeah, so maize, wheat, rice were the first plants to be domesticated, and especially the case of Natufians, which apparently they were the first that domesticated wheat, and they became from hunter-gatherers, they became agriculture, farmers. In fact, Yuval Harari says, this Israeli historian, he says that humans before domestication, they were very happy, they were very strong, they were walking and running through the African savanna, and then after domestication, humans became a bit sad and shorter and less healthier than before. So domestication of plants was key for human civilization, for human settlements, but also has a downside or side effects for the plants. For instance, well, not downside specifically, but a lot of side effects for the plants. So one of those, and it is quite common, for instance, for rice. For rice, the wild rice has a spontaneous chattering, while the domesticated rice doesn't have that characteristic to be able to pick it, to harvest. Also, we have seen this many times. So wild plants, the fruits of wild plants are not necessarily big or necessarily easily edible, but with domestication, what we look for is for plants that are easy to eat and very attractive for the consumer. So I will make this. It's not a test, but I don't know if you can recognize these three. These are three wild relatives of domesticated crops, and for me it's quite striking because I cannot say, maybe the one in the middle can be guessed, but the first one and the third one maybe are not that easy. So the first is watermelon. The second one is banana with seeds. You know that banana doesn't have seeds or are not viable, and carrots. Yeah. So this is the let's say the good side of domestication, but there is a downside or the negative side of it. Of this is the reduction in genetic diversity. So when we have the wild progenitor, we have a pool of genes that are in the whole population, but when we domesticate, when we humans domesticate, we select for a small amount of genes, or the pool of genes that the individual selected has at that moment, or had at that moment, no? So the domesticate, we wanted the selected gene for, I don't know, production, for height, for weight, whatever, but the neutral genes, you don't know which ones are lost, but many neutral genes are lost. Yeah. So I would like to talk about the process of domestication of one of the plants that I like the most, that the one that I work with in the PhD. So is the domestication of common bean. So the origin of common bean is being located in Mexico, in the Oaxaca Valley. And at the very beginning of research with common bean, the question was if was one single domestication event or multiple domestication events, because there are several evidences of possible domestication events. Nowadays, we know that common bean had two independent domestication processes, one in Mexico and the other one in northern Argentina and the south of Bolivia, west of Bolivia. No? The consequences of domestication, or the biggest consequence of domestication is, as I said, is the reduction of genetic diversity. No? And this is known since, I don't know, for common bean, This works for with Paul Goetz in 1998, where he demonstrated that the gene diversity in the wild relatives was much higher than that gene pool in US cultivars. This is not only for common bean, this is for all the known crops. So here we have maize, alfalfa, sunflower, soybean, and in here, in this column, we have the loss of nucleotide diversity during domestication. And except for one acorn wheat, most of our crops, modern crops, have lost a significant amount of nucleotide diversity. So for instance, 70% in emmer wheat, 62% in rice, and in common bean is not deception, as I told you, is 27% of nucleotide diversity. Then, when you compare, there are, I think I messed up a bit the presentation, sorry. Yeah, apparently in a few slides, I will have to repeat something, but what I want to show is that there are two main pools of common bean. One is Mesoamerican because of the Andes, because of the Mexican origin of bean, and the other in the Andes because we also had a domestication event in the Andes. And what you are seeing here is a structure analysis. The more lines, the more bars you see the more colors you see in the bars is more diverse. The less colors you see is less diverse. And this here is the Andean bean, and these here are the Mesoamerican beans, okay? And this MD means Mesoamerican domesticated, and this AD is Andean domesticated. So the bottleneck in Mesoamerica was very strong because the wild relatives of bean were more diverse. But as you can see in the Andean region, already the wild relatives were not that diverse. And obviously the domesticated Andean beans are less than diverse, okay? And then within the Mesoamerican and within the Andes, we have some domestic races of Phaseolus, Mesoamerica, Jalisco, Durango, Guatemala, and the Andean also has domestic races. And these domestic races were the ones used for further breeding programs, okay? The CIAT, which is the Center for Tropical Agriculture, which is in Cali, near to Medellin, is like, I don't know, 350 kilometers to the south of Colombia. They have one of the biggest common bean collections in the world, but not only bean, also cassava, and well, I don't remember, but they have also another crops. So if you need some accessions of bean, you can enter the website they have. CIAT belongs to CGR, and CGR is a lot of centers of research for food security in the world with different specialities, so rice, maize, potato, whatever. And then they have a freehold collection, a bean collection, and you can ask for accessions. They have more than almost 40,000 accessions. They even made a donation to the Svalbard in Norway to keep all these seeds safe and sound if we need it in the future. So with this background that I gave you, I would like to go into detail to the back to the roots concept. So at the very beginning, when I arrived, so Josh Reimakers told me, do you want to work with bean? Do you want to work with centers of origin? So your task will be to explore whether there is an impact of domestication of plants in the rhizosphere microbial community. At the very beginning, we established several objectives, one about the structure and composition of the microbial communities, and another one's about function. So I will try to, we will try to talk about many of these stuff. So what is the background and rationale behind this back to the roots approach? So back to the roots It's like playing with the words. It's back to the centers of origin, back to the wild relatives, and back to the microbes in the soil. So what we hypothesized at the very beginning was that a wild or native plant, despite they have low productivity, biomass productivity, and probably also in case of bean, the seed productivity will not be as high as a modern breed. They have high self-support, no? So when we domesticated or breed these crops, they became low self-support but higher productivity. This higher productivity is dependent on humans. So this man-made support to sustain high yields maybe has an impact, or maybe no. We hypothesize that has an effect on resistance through pests, towards pests and pathogens, emerging pests and diseases, increased abiotic stress, also led to soil erosion and depletion of nutrients and trace elements. So what we wanted with the back to the roots, and that's the rationale behind this project, was to reinstate microbial support systems exploiting beneficial microbes from wild crop relatives in their native habitats, okay? So I was talking a bit about microbiomes. Plants have many different types of microbiomes. They have microbiomes in the rhizosphere, which is the region surrounding the roots. We also have a microbiome in the nodules. The nodules are structures where bacteria are captured by the plant to basically to fix nitrogen, in this specific case of nitrogen fixation. We also have a mycorrhizosphere, which is all the fungi network that helps plants to get nutrients. And we have also an endosphere. The endosphere is the community of microbes which is within the tissues of the plant. So today we are going to talk mainly about rhizosphere microbes. Why rhizosphere microbes? Because the rhizosphere microbiome has proven to be a very important layer for the plant. Why? So this is the rhizosphere. This is a root, and the root is exudating, I don't know if that's the right word, a lot of compounds, carbon compounds. The plant can make photosynthesis, build carbon structures, and then release it through the roots. And a huge community of microbes can use those nutrients, but it's not only about nutrients. It's also about a niche. So as you can imagine, with these root hairs in this axis, all the superficial area, because of all these very, very dense root systems, is an ecosystem for microbes. And the rhizosphere is very important because the more diverse the rhizosphere microbiome, the more difficult for pathogens and pests to enter to the root. So additionally, we have in the rhizosphere the layer where the plant can communicate with mycorrhiza, with the rhizobial species, and with many other beneficial microbes. So the rhizosphere microbiome can be important for growth, can be important for nutrition of the plants, tolerance to abiotic stress, health. This is very important. For instance, take into account the suppressive soils. In the United States, you have beautiful suppressive soils in Washington, in the state, with all these wheat crops that are naturally protected by bacteria because of suppressive behavior of the soil. So that's why we are going to talk about the rhizosphere microbiome. But we are going to talk also about agriculture a bit, okay? So I am trying to put all the pieces together. So agriculture has a strong impact on natural habitats. For instance, this picture was taken by Lucas Mendes. Lucas Mendes is a collaborator of us, and he went to the Amazonian region to collect soils from these deforested areas to grow soybean. So he's been, Lucas Mendez and his group, they have been monitoring how the microbial communities changes due to agriculture, and obviously due to the newcomer, which is soybean. So in the back we can see the Amazonas forest without being deforested, and here you have a lot of soybean just sown. So there are two main questions here about, obviously, about how the plant modifies its environment in the rhizosphere, a wild or a mother plant, but also how the agricultural management can have an impact of soil. So I have several questions that I would like to try to address in this today. So, do wild plant harbors more diverse and stable microbial communities as compared to mother plants? How is the composition of the microbiome in soils from centers of origin? But in here we have to be very careful, because soils, we have to make the distinction between agricultural soils and soils without being disturbed. And these more general questions about the process of domestication and how it affected plant traits for plant microbes interaction. So at that moment, we started with this strategy. I will go step by step. So the first step in this strategy is to select soil from the centers of origin and plant material, wild, land races, and mother varieties. Okay, so we said, let's take those soils and these plants to the greenhouse to try to control most of the, well, as much conditions as possible. Then assess the rhizosphere microbial community. We have done culture-dependent approaches, but mostly I will talk today about culture-independent approaches. And the fourth, but the most difficult one, is to reinstate beneficial partnerships in mother cultivars. And for this, we have been thinking about marker assist selection and plant breeding, but so far we have only come to this third step. The fourth step is still elusive. So this is why I told you that I mess up with my slides, but I will go straight to this one. So why I talk about bean? Because it's my model plant. Okay, so Colombia is mainly from the Mesoamerican gene pool. I told you that common bean was originated in Mexico. Then common bean made a journey through all Central and South America, and then three different pools were settled. Colombia belongs to the Mesoamerican gene pool. So knowing this, we went to see it. We had the assistance of Dr. Daniel Debuc, and a biologist that he was a very good biologist, Orlando Toro. He passed like 7 years ago or something, very nice person. And they help us to select wild materials, land races, and modern varieties. Once we did that, the first thing that we did was something very basic. And I think when we are talking about wild relatives and mother relatives, you need to know how modern are those and how old are the wild, no? So you need, at least what we thought at that moment is, let's sequence with a technology, the name is DART, with markers. Let's make a survey how diverse are those genotypes. So we sent for this genotypification strategy that I just mentioned, and we found that from the 8 accessions they gave us in SEAT, only 2 were real wild common beans. Okay, and what you are seeing here is the more lines, the more red lines or green lines or blue lines you find is because there are more homozygous regions. And homozygous regions means that probably they have been bred, or they doesn't have that much diversity. They have the same genetic elements once again and again. So what we found is that one of the accessions, and the name is M1. They said that it was a Y relative, but according to our analysis, was more related to a mother variety. So for this study, we had 2 real wild plants with really low amount of homozygous regions and 5 modern varieties. And very interesting one, this landrace, only one real landrace was this one, and is more related, is not related with the modern nor with the wild relatives. So we left it alone, okay? And we also measured something that for us was really nice, was the specific root length. And we find out that supporting this data, the wild relatives have a higher specific root length. So the roots go a bit deeper, yeah, as compared to the modern ones and even the landrace, okay? So a phenotypic trait was in accordance with what we observed with the genotypification. So for the microbiome profiling, what we did? So we did and pick up soils. Colombia, and this is very important, and I don't want to be misinterpreted, Colombia is not the center of origin of common B, but is a center of diversification. So we have many different types of land races and many different types of wild relatives of common B. So we went to a region where traditionally common bean has been grown, and this we called the agricultural setting. And we also went to a region where most of the wild relatives in Colombia have been found, and we also collected soil in that part. So the microbiome profiling strategy was more or less like this. We collected the soil, we sieved the soil, we disinfected and germinated the seeds, and then we made pot experiments in the conditions where common bean can grow easily, no? So all these common beans were voluble, so we needed to put a structure for them to climbing beans, and then we collected the roots, we checked the roots, we collected the rhizosphere soil, and we extracted RNA and DNA. More or less, this was the strategy. We sent for sequencing the V3-V4 region of the 16S. So when we talk about 16S, we talk only about bacteria, okay? Why we went for bacteria? Because it's the most abundant portion of the microbiome and probably the most diverse. Also, I will show you that we have also worked with fungi and protozoa, but in this study, we only worked with 16S data for the rhizosphere. So I will jump straight to the results. So this plot in the left is showing bulk soil. This is normal in the microbiome studies with plants, with rhizosphere or with the endosphere, that we take into account the bulk soil. So the bulk soil is the soil without roots. And you can see that they group together more closely as compared to the other samples. Each of these is the rhizosphere of a plant, and the red ones are the modern ones, and the green ones are the ancient ones or wild ones. So what we saw here, this is with soil and this is without the soil. What we saw is that the rhizosphere samples from wild relatives tend to cluster together, and the modern samples tend to cluster together. And there is a differentiation between wild samples and modern samples. So for us, this was amazing, mainly because the variations that the genotype of a plant can exert in the soil is no more than 5 to 10%. And we saw a strong effect, more or less of the 7%. We wanted to know which bacteria were explaining this behavior, and this behavior was explained basically because two main phyla of bacteria, bacteroidetes and actinobacteria, okay? So what we observed is that in red, you have those bacteria that are enriched in the mother varieties. And in green, we have those bacteria that are enriched in the wild relatives. We had a lot of bacteroidetes, especially Chitinophagasia and Cytophagasia, two families from bacteroidetes that were really, really abundant in the rhizosphere of those wild relatives of common B, wild common B. And these actinobacteria were extremely were more enriched in the mother varieties. So following this, I remember that when I was doing these experiments, I read this paper from Davide Bulgarelli and the group of Paul Schulze-Leffer in Germany, in Cologne, and they found the same. So when they explored the Ordeum bulgarium as barley, they also observed that bacteroidetes was most abundant, was the most abundant phyla or phylum in the wild barley, explained by one of the families. This family is Flavobacteriaceae that is very common in Europe. This family of bacteroidetes. So we were very happy to find this, and we wanted to follow this thread, no? So we took, at that moment, we are talking about 2017, we downloaded all the available data from all the papers that were published until that date regarding the microbiome of wild plants. Arabidopsis, sugar beet, barley, lettuce. We had more data, but only a few sequences could be used because of many different filters we settled. But at the end, what we observe is that there is a huge effect of soil. So what you can see here is, for instance, Lactuca sativa. So these and Lactuca areola, these are from the same experiment and were done in one type of soil. So that's why they are closer together. So the first force or how to say this, like the soil is the biggest force for the microbiome. But we did an analysis, a differential abundance analysis applying the same model. The model is a very nice model, generalized model that help us to find this trend. Bacteroidetes was the most abundant bacteria for all the wild relatives we assessed. So in Ordeum bulgare, spontaneum, in Lactuca areola, which is the wild relative of Lactuca sativa, obviously in our study, and we also took into account Cardamine hirsuta versus Arabidopsis. Cardamine, let's say, is the oldest of this group of species, and we were very intrigued and very excited to see that Bacteroidetes was more abundant in wild plants. So we proposed this hypothetical model in which wild plants are more abundant in bacteroidetes, but basically because in the soil there are several things that are happening. The specific root length is higher, and there is less exudation of simple sugars. Mother varieties exudate a lot of simple sugars, and also pH, phosphorus, nitrogen, and the water supply are less abundant, and the pH is lower in the native environments of these wild plants. So, and also we propose actinobacteria and protobacteria were higher in modern cultivars. This is a hypothetical model that we propose and is still to be confronted, no, or validated. So we were very interested in this group. Bacteroidetes is a gram-negative bacteria. We have a lot of bacteroidetes. Humans, we are full of bacteroidetes. In soil, as I told you, there are also several genera. In humans, the more high fiber diet we have, the more bacteroidetes we will have. And in the soil, the more complex biopolymers you have, the more bacteroidetes you will have. They have a high diversity of carbohydrate active enzymes, and they are associated with traditional diets. These studies I wanted to show you, in which uncontacted Amerindians, children for rural Africa, and the Hadza hunter-gatherers, they have in common that their proportion of bacteroidetes is higher as compared to the modern or westernized people. Yeah, so for us, this parallel was very interesting. So plants, modern plants, lost their bacteroidetes, and modern humans are losing also their bacteroidetes. So this is something that for us was very interesting. But, and I will try to rush a bit. This is dependent on the soil effect, because what we observe in this agricultural soil that I mentioned, so the green ones are the wild, the red ones are the modern. We can see the grouping in here. We lost that sign in the native environment. Okay, so the bulk soil is still completely different from the rest of the samples. But then when we analyze the rhizosphere microbiome of those samples separately without the agricultural soil, we don't see the pattern that we observed at the very beginning. So we wanted to analyze why we lost that wild relative effect. So first, what we first do was a core microbiome analysis, and what we found is that the core microbiome between both soils is not necessarily the bacteria that are more abundant for all the accessions independently of the soil. I explain myself a bit better. For instance, the native environment had a lot of Burkholderia, yeah, while the agricultural soil doesn't have that amount of Burkholderia. Just to talk about one of the genus, Rhizobium. In the native environment, the number of nodules was really low, while in the agricultural setting was very high. So we had a lot of Rhizobium. Also, when we analyzed the co-occurrence networks, so what we are seeing here is the interactions between the bacteria that we see. And what I want to show you is very easy. Well, something very simple. I think we can observe it. So we have every number here is a niche. Imagine it as a niche. I don't know what kind of niche, because we don't have any clue about that, but we can speculate. So all the green connections, all the green dots are bacteroidetes. The blue ones are proteobacteria, the red are actinobacteria, and there are several other colors. But what we saw is that bacteroidetes has a different niche in the rhizosphere in the agricultural soil. Also, the rhizobium has a completely different niche. It doesn't mind for rhizobia, doesn't connect with the rest. They are just one group doing their stuff, no? And if you see the number of connections, and there is only one negative correlation, and is between the group of bacteroidetes and the group of proteobacteria. This network is quite simple. The number of edges and nodes is very low. And we concluded that maybe the agricultural settings is reducing the complexity of the ecosystem, while we have all those the same accessions growing in a native environment. And what we see is a jungle, is the struggling for survival. So we have a cluster here of bacteria that likes to live in environments that are oligotrophic with low amount of nutrients, and they are fighting against these ones which are proteobacteria, mainly proteobacteria that likes to grow very fast with a different kind of strategy. So what we concluded from this is that agriculture simplifies the environment for the plants, and most of the time what we see is a less diverse number of habitats or niches within the rhizosphere, while this is not happening in the native environments. By the contrary, the ecosystem is quite complex. We saw exactly the same in one experiment with wheat. So what I have shown so far has been done in common bean. So we moved to wheat in Brazil, where we planted several land races of wheat, several modern cultivars of wheat in an agricultural soil and in a forest soil. And what we observed was exactly the same. Land races, well, not exactly the same, because in the agricultural soil, we saw that the land races have more connections. And in here, we added layers of complexity, because in here we have bacteria, we have protozoa, and we have fungi. And we have a lot of negative connection between protozoa, bacteria, and fungi, obviously because protozoa feed from these microorganisms. In the forest soil, the same. Land races holds a more diverse, or at least more interconnected community, while the cultivars doesn't do the same. Unfortunately, we couldn't do the same analysis here because we didn't have enough number of accessions to do the analysis per wild and mother. In this experiment, we had a good amount of samples, so we were able to do it. So some take-home messages: bacteroidetes is not a general observation for other plants in the last publications I have seen. At that moment in 2017, we were very excited because we found it in many of the studies, but many studies have shown up the last, I don't know, 3 years, and sometimes it shows up and then we don't see it anymore. It's a very interesting group, and I also wanted to stress that in agricultural settings, soil is conditioned. So while relatives or mother plants will grow more or less with the same conditions, because the soil is already modified to, I don't know, to support certain type of microbes, okay? And lastly, to finish, I would like to show you some of my recent work. I am working now with a bacteria which is devastating for banana and plantain crops in Colombia and in the rest of the world. These exudates that you see here are from this bacteria. And what we are doing now is to apply bacteriophages before the bacteria and after the bacteria, trying to see if these bacteriophages can stop the spread of the bacteria. But what is the important thing here? We set this experimental plot in the north of Antioquia, near to the coast. And what we want to do is to evaluate whether these bacteriophages have an impact on the rhizosphere microbiome. I mean, whether the bacteriophages affect other bacteria in the soil or affect the whole composition of the microbiome. And for that, we have been working the last 2 years with several students. We have put a lot of effort on this. Is a pipeline to work with metagenomes, and we call it metabiome. So one of the biggest problems in metagenome projects is what to do with the reads. There are many programs, there are many ways to analyze those data. So what we propose is a modular pipeline in which you can use any of these modules. You can extract the 16S, you can extract the functions, the taxonomic profiling, the taxonomic binding. Well, you can do a lot of things with that metagenome, I hope in a couple of months we have all the documentations and we can send for you if you want to use it. So with this, I would like to acknowledge a lot of people from many institutions at CIAT and University of Antioquia in the Netherlands. Well, this work was done during 6 years of work in the Netherlands, and many things to say thanks about, many people to thank. So if you have preguntas, I will be very glad to reply, and thank you for your attention.
Thank you so much, Juan. Yeah, if anyone has any questions, I mean, you can put them in the chat and I can ask them, or you can unmute yourself and ask them, whatever you're comfortable with.
I have a question. First of all, Juan, un placer conocerlo y buena presentación. Estuvo muy interesante. Basically, what I take out of your project is basically not only the importance of the genotype determining the bacteria associated with the wild relative, but also the importance of the environment as well. In Mexico, where I also work with wild relative, is easily like when you talk with people, you get to the conclusion that wild relative are basically endangered. With the years, the population are decreasing rapidly. I'm wondering, in order to also conserve those microbes that are associated with the wild relative in the native environment, what is the conservation status of the wild beings in Antioquia where you were working, other than just like what it is done everywhere in the world, just like by conserving the seed and by conserving the genotype, but the conservation status of those plants in the actual like wild environments?
Well, thank you for your question, Jorge. I think it's endangered because the habitats are very threatened by human activities. We don't even know how much wild relatives we still have in the mountains. There is a region that is near to the to the border with Panama, where common bean came to South America, where we still have a lot of forest. So we don't know. I don't really know if someone has made a monitoring of wild relatives in that region, but I hope that because this is so known and so untouched, there are still a lot of wild relatives of bean in there. But in the Andean portion of Colombia, I can tell you that the Andean region is the most populated and the most degraded. So I will be a bit pessimistic about how many wild relatives we still have in the wild.
Gracias por la respuesta. Thank you for the answer.
Yeah, gracias a vos.
Sí.
Vic, I think, has a similar
gracias,
question.
Juan.
Yeah.
Gracias. Muy interesante.
Gracias.
I have a question related to this association of diversity with the specific genotypes of plants. And I'm wondering if you took into consideration, similar to like the environmental effects of maybe layering the no-till or low-till versus the high tillage systems, because it looks like possibly that it's the tillage systems are just knocking back this diversity and not allowing kind of evolutionary processes to move forward to allow for diversification. So if you looked at no-till systems, would you see eventually that diversity to build up again?
Yeah, well, that's as you saw. Thank you for the question, Vic. As you saw, we took the soil and we completely took its structure away. No, so all the aggregates were completely destroyed. So probably that has an effect because within the aggregates we have microbes within the aggregates. And maybe when we destroy those aggregates, we went against many of the microbes that lives on those aggregates. So you are right. Probably if we try this type of experiment in a more natural setting with a soil with a better structure, probably we will have a different outcome. But yeah, for me it is hard to say whether will be very different from what we found in a non-tillage soil.
Yeah, just to clarify, I'm talking about where you sampled the soils from. So you sampled them from agricultural soils that had high tillage, or did you sample them from soils that were perhaps low tillage or no tillage systems that maybe you would actually sample a greater diversity from those?
Okay. Sorry, I didn't.
Or if that would be a possible avenue to research, if you think.
Well, the native soil was from on the stored land. So we took all the litter away and we took the soil just as it was in that part of Antioquia that we presume where white relatives entered to Colombia. The agricultural setting in here, in the part where they grow common bean, is not common to tillage with machines. No, is the people with the asadón. I don't know the name of that tool in English, and they do it by hand, but not really deep because it's a lot of work, you know. So what they do is just to remove a bit the soil and for to sown the bean seeds, they use a tool that the name is recaton. The only thing that you do is a small hole and then you put the seed. So I would consider no tillage, but really low tillage soil. Yeah.
Cool. Yeah, that answered my question. Thanks.
Yeah, good.
Excellent talk. Really enjoyed it, Juan. This is Jean Harris.
Thank you very much. Thank you. Very much.
And I was wondering, you touched a little bit at the beginning on the idea of resiliency with these native microbiomes. And I wondered whether you have, I was really interested in those co-occurrence networks. And I wonder what would happen if you added some kind of abiotic stress. You know, would those simplified networks break apart more? Would you see more reduction? And I'm curious what would happen with those massively connected co-occurrence networks with the wild soil. I wonder if you're kind of thinking in that direction at all, because I'd be really curious.
Yeah, well, I think one of the abiotic stresses that can have the biggest influence on these co-occurrence networks is pH, because we have seen that when we observe networks in low pH, I have some data on those, the network is more complex. So the competition, I think, is higher in those environments. The resources are too low and so unavailable, many of the nutrients, that they fight very hard for it. And that's why the other side of the coin is the agricultural settings. In the agricultural settings, pH is high, the water availability is high, so the complexity is lower and lower and lower. I think pH and water content, because the other ones, bacteria can do many of the things, can solubilize phosphorus, they can fix nitrogen. So I think the most important ones are pH and water. Yeah.
Thank you.
Yeah, you are very welcome.
Well, thank you everyone for staying on for a little bit and asking so many wonderful questions. We will now ask everyone but grad students to leave. Any grad students who want to stay on, Juan has agreed to have like a 20-minute session with us after to talk about any specific questions we have about his work or our work, and also just career development type stuff. So thank you everyone, and
thank you very much for listening and for the invitation. Hope to get to know you in the future.